So today's Ento Live webinar is about a subject that I've been dying to hear about for a number of years, and it's the Rothamsted Insect Survey. So I'm not going to go into too much detail about what that is, because I'm sure James is going to do that. And I'm going to hand over to James Bell firstly, then we're going to listen to Yoann Bourhis later on with some of his research that's related to the programme as well. So over to you, James.
Thanks very much, Kieran. Thanks very much indeed, and welcome everybody to the talk about the Rothamsted Insect Survey. So I could, to be honest with you, have all day talking about insect survey. After all, it started in 1964, and it has had huge impact since that time. So naturally, there are things I'm going to miss out, and I apologise to anybody who may have collaborated with the survey who hasn't seen their work, but I think you probably understand. Now, throughout my talk, I'm going to be referring to the website, and you can link to the website insectsurvey.com by scanning, if you can on your PC, the QR code on the left-hand side there. And as I talk through, going through the website at various moments through my talk, you can follow me too and see some of the services that we provide. I'm delighted to say that I'm going to be joined by Yoann Bourhis, and Yoann is a postdoc with me on the DRUID project, which is Drivers and Repercussions of UK Insect Declines. And he's going to talk about a subject probably very close to your heart about how you can use citizen science data to predict new distributions of insects using their traits or their life cycle understanding and also environmental drivers. So without further ado, let's get on with this. So if I were stuck in a lift with a policymaker or a funder and they wanted a simple, very catchy understanding as to what we do, I'd probably tell them that actually the Rothamsted Insect Survey makes your weekends better. And that is because all the things that you see here, good or bad, the Rothamsted Insect Survey has a role in monitoring and surveillance. So on the left-hand side, you can see my local pub, the Black Inn in Leighton Buzzard. Now, like hundreds of pubs over the whole of the UK, they serve up real ale, and real ale is flavoured by hops, and hops are grown in both Kent and Hereford principally, but also they occur in the wild in field margins too. Now, there's an aphid that feeds off hops, and that aphid actually reduces the quality of those hops. And so we monitor that and send information out on furrowed on the hop aphids to the wider world. Now, you might wonder what on earth a kebab is doing inside the middle of this picture, but actually, it turns out that we do a lot of monitoring in this space. Of course, you kind of associate our work with host plants, but there are also animal hosts that we collect, such as Culicoides midges that feed on sheep. Now, sheep is a principal component of a meat kebab, and I'm accepting that we have vegetarian and vegan kebabs too. But blue tongue disease is vectored by Culicoides midges, and this reduces their health greatly. You can see lettuce there in the picture there too. Now, lettuce is a really interesting crop. I'm going to talk about many crop plants that are affected by aphids, not through their feeding activity, but through their viruses they transmit. But the lettuce aphid, researched at Rothamsted Insect Survey by my former PhD student, now postdoc, Dion Garrett, he was trying to understand Nasonovia, the lettuce aphid that feeds on this, this plant. And what he found out was actually in our traps, it occurs at low density, but in the field, it's a contamination issue. And we all strive for higher quality food, but at great cost sometimes. And lettuce is a perfect example of that. So if Nasonovia, the aphid, occurs in a lettuce head and it's seen in the supermarket, then the supermarket is not pleased with that. So contamination is a real issue. And what actually happens in the field here is that if tens of heads are detected by a crop consultant in the field, the whole crop will go. The whole field of hundreds or even thousands of lettuce will just be removed. So that's as a consequence of us demanding high quality food, not accepting a little bit of aphid protein, if you like. Now, good or bad, sugar is in all our diets. And recently we learned that whilst we don't have the highest sugar demand across the world, we do consume on average as adults 34 kilograms of sugar per annum. And sugar features in fizzy drinks, features in cakes, even features in bread. And our work on sugar beets to understand how we can mitigate against prophylactic use of insecticides, the main mission of the Rothamsted Insect Survey will form a topic in my talk a little bit later. Now, some of you can see a glass of wine there and wonder what on earth that's doing there. But wine in its broadest context, including things like fortified wine such as brandy and sherries, all are affected by an aphid called phylloxera. And phylloxera really damaged the French industry in the 1850s. And in the 1850s, a third of French people were employed by the wine industry. And this really came to a bad end because phylloxera, a root-feeding aphid on vines, destroyed cognac, but actually serendipitously created a new region called Armagnac, which you might be familiar with now. And that little aphid allowed the Armagnac to grow, and also a new railwork and new jobs for those people in Armagnac. So pests can be good and bad, mostly bad, but also some of our work, I should say, informs what we do with the BTO and RSPB, where aphids are often included as food as well as moths. All around this subject, of course, is our work on moths, and you're going to hear a lot about that a little bit later. But moths, even in agriculture, are studied by us. We're looking for non-target effects, and we're looking for biodiversity changes, all of which you'll hear about in one of my topics a little bit later. So let's move on now. Now, we've just won the next 5 years of funding with BBSRC, and these are the topics that we've actually got to cover over the next few years. And those highlighted in bold here are those things I'm going to talk about in more detail. So I'm going to talk about emerging threats, and I'm going to talk about our work on deep learning tools to automate insect ID. And I'm sure for many of you, that's a thing that's already familiar. You might have, for example, Obs Identify, an app on your phone to automatically identify many charismatic insects. But with deep learning, it's really struggling with those small blobby things like aphids. We also look at community differences and show how moths in particular change over time. And our forecasting and surveillance will feature in my talk about sugar beets. I want to invite you throughout the talk to some of our services, 2 of which are Digital Sample Archive, where you can come to the survey and request samples. The samples that you can request are from a source of 100 million insects that you can actually study with us. And if you're not satisfied with that, come and dig into more than our 50 million records of aphid and moth and other insect data through our data warehouse, all of which I'm going to talk about a little bit later. So just to make you aware, and I'm sure many of you are aware at least of one trap network, we have 2. The suction trap network I'll talk about shortly, which is effectively an upside down Hoover, and also the light trap network, which is focusing on capturing nocturnal insects, particularly moths. Now, together, when you combine the science that they do, we cover a huge area from theoretical ecology, trophic ecology. In fact, I'll show later how Hannah Romanowski is doing work on bats and insects, and biodiversity conservation, for example, probably close to all of your hearts. But I also do fundamental research on insecticide resistance and how viruses are transmitted and what the threat is to agriculture. And we also study things associated with climate change. And unbelievably, since 1964, climate change has emerged as a topic, but at the time, it was not known to any of the staff at Rothamsted who set up the survey that climate change was actually a thing. How times have changed. So let's start with the upside down Hoovers, or the 12.2 meter suction traps. And you'll see these dotted about the country. And the map you see on the right-hand side shows you many of the traps that we have in the country, all the way up to Scotland and all the way down to Devon and beyond. And some of those traps are still running, but our best long-term running sites are the ones in red, and particularly those that started in 1964, such as Rothamsted, where I am now, and Broom's Barn, which is in East Anglia. Effectively, what happens with this suction trap, just like a Hoover, is that there is a suction at the top of the trap where the point is, and that draws down insects up to a size of around of a ladybird, but more commonly it's targeted at smaller insects like aphids. But we get a whole range of hoverflies, and you'll see later the different groups. such as flies or diptera that feature very heavily in the work that we do. So if you want to know more about the history of the insect survey, please do go to my paper in Journal of Animal Ecology. It's open access, so you can click that link or go to it using the title quite easily. I should say that when we collect the sample at the bottom of the chamber, we then take it into the lab, and amazing stuff happens. I just want to move on and just post a very quick slide about the Rothamsted Insect Survey suction trap network and how it's spread throughout the world. We were the first to create that, work by L.R. Taylor and C.B. Williams and others showed that actually this was a really useful tool for agriculture and also for sampling insect migration generally. And countries throughout the world have copied our design, and some of them vary in style and height, but essentially they represent the same components. So you could go to China, you could go to Canada, you could go to the US and find similar networks like ours. So at Rothamsted, across England, samples come in, and our expert team who are trained in aphid ID are there to identify the 400 aphid species that they may encounter. I'm going to talk a little bit later about how to identify an aphid, and it's quite different to anything else. If you know anything about insects, put it on the scale of staphylinid beetles, because they're pretty hard, and so are aphids too. So we train our team. We can't really readily find aphidologists in the citizen science area. And if you look on NBN, you won't see many aphid records all the same. Confirmation that really aphidologists are rare. But we have an expert team there who identify the aphids and then split them into their species components. Remember, we're talking about 400 species of aphid here. And also we find a new aphid every other year. So really quite a challenge. They all look pretty much the same, small, brown, and blobby. So nothing charismatic like a butterfly or a moth to see here. They then make their way into an allocated tube, one for aphids, another one for non-aphid fraction, as we call it. And Chris Shortall leads that team. You'll see later some of his work. And here, what we're doing is partitioning that sample so we can get to that quite quickly. It then goes down to the archive, and the archive is a place where those samples are stored. And I'm going to talk about that in a moment. But before I do, I want to say something about how we transmit that data that we record in the lab in quick time, not real time, but super quick, back to crop consultants in the field. Now, the objective here is to reduce the prophylactic use of insecticides. And those people who are a little bit older, like myself, might remember a book by Rachel Carson called Silent Spring, all to do with the prophylactic use of DDT that was causing eggshell thinning. The people at the time, the staff here at Rothamsted, were really concerned by that. And they thought that if they communicated the threat, then people, the farmers, the growers, wouldn't prophylactically spray. And that's proved to be true. And farmers are much more aware now of the danger of doing that. And we like to think that we've played a huge part in that. So if you go to our website, you click aphid data, and then you look at the bulletin, the aphid bulletin, you can see these 4 regions across the country. Now, the northern region in yellow is covered by SASA, our partner organisation in Scotland, that essentially does the same work as we do for 4 traps, 4 suction traps spread out in the agricultural sector across Scotland. But we're confined to England. And here what we're looking at is the purple area. I've just chosen one at random, happens to be the northern England, where we have 3 traps. And then if you click the crop type, in this case we've chosen cereals, we'll see the 3 aphids that are affected yields in this area and the traps in that region. And these completion graphs, if you do any running or any fitness, you might be familiar with these, but they tell you about the performance, if you like, in inverted commas, of the suction trap. So, for example, Newcastle rose grain aphid has recorded about 1/3 of its expected catch for the year. That's the highlight in yellow. The weekly catch for this week is number 3, and we've got 2/3 to go. But what we do know is that since the week before, that trap number is going down. Now, sometimes these numbers can go into the thousands, 2,000, 3,000, particularly in September and October when we see high activity in these cereal aphids. Now, I mentioned at the very beginning about the kebab, and I just want to explain a bit more detail about the holding material for that kebab, which is, of course, a flatbread. Now, flatbreads, of course, are made of cereal. Unless you don't eat bread, you'll come across bread in some shape or form in your average day. And bread has been affected by these cereal aphids. They transmit a virus called BYDV, barley yellow dwarf virus. And typically that has a yield effect of about 10%, reducing the seed heads through the virus transmission. Now, warmer winters are extending the transmission period, which means that next year, when we are expected to have a super El Nino effect, i.e. a very warm winter and a super hot summer, we're expecting these cereals to be greatly affected because the migration of the aphid will be early, and I'll show that graph a little bit later. So the kebab, just to complete the story, is affected by 3 aphids. It transmits the virus and yields are reduced. And particularly in bad years, when we haven't got resistant varieties, that can rise to around 40% of loss. Now, I talked about the archive. And the archive essentially is downstairs, 100 million insects in our basement. And I mentioned about the suction traps, which is largely providing the material for the archive. And these are about the size of a ladybird or a small beetle. And there could be chafers, ladybirds, lacewings, and vectors of disease, human disease and animal diseases, as you see on the right-hand side, as well as hoverflies too. And we have the data and also the samples for those. So if you're interested in the genetics or you want to revise the taxonomy, come and see us. I'll show you how to make an appointment in this talk. So I just want to explain some of the uses of the archive through the PhD students that I've had working in Insect Survey in collaboration with others, of course. Now, Chris Shortall is still with this survey. He's a senior entomologist at Rothamsted, and he studied the oilseed rape pest, the Melagethys. And what he did is he went through the archive to recover the incidence of that pest, and then linked them with the amount of oilseed rape in the environment across Great Britain to show that really it wasn't being regulated by that pest. So the pest was actually increasing and decreasing irrespective of what oilseed rape was doing. And Dion, again, he's a postdoc with the Insect Survey, and his work during his PhD was just about lettuce aphid that was greatly affecting the contamination of lettuce, where we saw, and I talked about how whole fields were removed just because of contamination issue. Demetrius is at Newcastle with Darren Evans, and he does some great work in collaboration with us, looking at how you can preserve specimens without ruining them for others by taking small body parts to have meaningful recovery of DNA. And he uses metabarcoding technology to understand how that could happen. And Hannah is in year 3 of her PhD. And just to give you some examples of the research that we do, she's working with bats. She's a bat specialist, and we're training her to be an entomologist, which she's thriving at, and to understand how bats and insects are related. Are bats' numbers strongly related to the availability of their food? And that's something that Hannah will tell us all in the next few years. I just want to say an advert to anybody who's interested in moths. In the archive, we have a special collection of moths. This is unusual because we don't normally keep them, but we've tried to preserve a great many moth samples. So if you want to use that facility, do get in touch with us and use the appointment facility. I'll show you a bit later. So when we take the suction trap catch, of course we get aphids. What else do we get? We get Diptera, Coleoptera, Hemiptera, Hymenoptera, Thrips, Thanatoptera, and other insects, as well as, I'm delighted to say, as someone who did their PhD on spiders, arachnids. And I always am delighted to see now and then when I drop into the lab, 1 or 2 pseudoscorpions. Probably a nuisance insect, if I can call it that, is the thrips. There's so many of them, we can't identify them. And they represent, whilst a small biomass, a huge number of insects that we record. So again, if you see anything on here thinking, do you know what, I could use the archive to go and look at some family of Diptera, do get in touch with us. So here we are. This is what it looks like. We're now downstairs in the archive at one of the samples. You'll see that a well-preserved sample from 1978 is in a box, and we're now digitizing that so we can get faster recovery. And if you want to know about that, contact Chris Shorter, who will tell you all. But suffice to say, it's an open access resource, and we invite you to come along. Now, of course, we've got the physical sample of those insects, but we also have got the digital sample too. And so if you want to have the data that we record, and admittedly, there are some groups that we don't represent. For example, if you wanted anything about thrips, we really don't have the data for that. We might just score them a lot now and then qualitatively in one of our RIS remarks, but otherwise, not really. But most of our other insects have some data at least that you can check in on. So we'll see the process here. We've got the lab, as I talked about, then that moves to the laptop, as you'll see that person on the right-hand side of the screen. We then clean that data, and it enters an internal database called Paul that stores all data from 1933. Now, what I didn't say at the time was there was a small activity with the light traps from 1933, stopping in between the wars to record some of the moths at Rothamsted. Now, that is an unusual data set, not common, but it does show we've been around for quite a long time. Paul talks to our warehouse and sends the data from 1990 onwards on a daily basis to you, the client, and you access it via our portal, and you can then get a data set with its own DOI that you can reference in your papers or in your work that will have the catch data and the trap metadata, everything that you want to know about trap precision and recording. And from that, you can actually require different types of data or call different types of data from the warehouse, including just, for example, daily totals, or if you wanted annual totals, or percentiles, if you're looking at changes in phenology, for example. So again, I'll show you how to do that. So we're now on the Insect Survey webpage at Get Data. So we record the bulletins that I talked about earlier, but everything else that comes from the suction trap that we are able to identify, you'll see in RIS remarks. So click that link. You'll see lots of reports over the last year and many years besides on the qualitative counts of many of the most interesting insects. So we might report, for example, a high number of beneficial insects and break that down by hoverflies and ladybirds, for example. So that might be useful to you in your research. Now, the data warehouse, you want to get some data, click this link. You'll then take them to a portal where you'll create a login. And from that point, you can then download and use the data. I encourage you to, if you have any problems, to contact Alberto in the first instance, who is there on the portal, and or Chris, who can help you acquire the data you want. Now, I should say that if you want really long-term data before 1990, you need to get in touch with us, because it then gets quite complicated. We're happy to provide it to you as a data set, and there's no requirement for you to include us as co-authors unless you want our expertise. So really what we're saying is, here it is, come and use it for your own research, which is a great opportunity for anyone really, particularly those PhD students who may have been affected by COVID or want some extra data to bolster some of their research. So check that out. And the sample archive, you want to make an appointment, then you just click this link, you then fill in your request, just describe very briefly what you want to do, and I'm sure we can accommodate you. So now moving on to the light trap network, and some of you will be familiar with the light trap network, but essentially it is one of the longest data sets in the world. We began recording in 1933, as I described rather, and it goes up to the present day. Typically in any one year, we have between 70 and 80 traps operating. Now, of course, traps start and they stop, but typically they last at least 10 years, and some of them nearly 60. So there's a wealth of data there for you to use, and you can request data for a single species or a whole group of species at the site or across the network. It's all your choosing, really. But what you're looking at here is the science behind the flight to light response. So we turn this off at turn this on at dusk and off at dawn, and it's there to catch nocturnal insects. And as you might expect, moths, of course, are attracted to that light, and they're collected in the chamber below. But other things too, like winter gnats, are also collected. So whilst there's only a small bycatch, there's a bycatch all the same. Now those moth samples typically don't make it into the archive because they rot very easily. And also it's not often the case to record anything other than larger moths. So the micro moths, unless they're a special case, for example, a couple of years ago, we had a request for the beet moth to be recorded across the network because it was migrating over from continent. Unless it's something like that, micro moths generally aren't recorded apart from a couple of species. Now, what happens to our data beyond you as clients using it? Well, we collaborate with Butterfly Conservation and CEH and others to really try and deliver impact from the data we collect. One example is the State of Britain's Larger Moths 2020 report. And if you want that, you can download it from our website by going to Moth State. So in that report, you'll see some trends on the abundance of moths. And as many of you might know now, moths are declining, and yet nationally that's the case. And there's an interesting picture between the north and the south, with the south declining faster. We can make things more complicated by looking at the non-linear trends. And not surprisingly, there are sometimes good years and bad years of moths. And there's a lot of science, a lot of applications that you can explore at Rothamsted that are trying to unpick what's happening here. Now, if you go onto the moth data and then look for trends, you'll see these graphs. And these graphs are work in collaboration with CH to look at 432 abundant moth species caught between 1967 to 2016. And what you're looking at is really quite a complicated model to adjust for some imprecision in the trapping and also missing data, for example. And that's quite a technical explanation that I'm not going to go to here. But essentially what we've done is provide the most robust trend model for individual species. And here, typically, you can see that the moth is in decline. I'm going to show you a little bit later, just to bring the spirits up a little bit, about some of the moths that are actually increasing. But do have a look at that. If you've got a particular moth species you're interested in and want to know the overall trend, this is the place to go. So I'm now going to move on to 3 topics. The first one is all to do with identifying aphids using machine learning, or actually in particular, deep learning. Now, this could apply to a parasitoid, a very small staphylidid, any insect you can think of that is lacking charisma. And I don't want to be rude about these insects, but they ain't the prettiest and they don't certainly have enough color there. But models really struggle with identifying them because there isn't too much information which they can grab hold of and understand to actually make a precise prediction about what species it is. So let's go through that. I have to say that there are just a tiny minority of the 600 or so aphids that occur in the UK that are in some way charismatic. And here they are. And this is because they may have color differences on their wings. They may have bimodal color patterns on their body. They may have waxes on their chitin, or they might just be super large. And our largest aphid occurs at Rothamsted and elsewhere just about now in autumn at the base of willows, called the giant willow aphid. So get really excited about that if you see a willow and you get that aphid, because that's the biggest you're going to get in the world. But for the rest of the species that we record, they are small and blobby and brown, yellow or green. And that's a problem, because what we need for computers to work is to absorb a new taxonomy that's not necessarily based on their color. Now, when we train scientists, we actually give them specific characters to look at. And these are the rhineria on the antenna. So they're the sort of sensory glands that are trying to understand the chemical environment around them, as well as what I call to the general public, the exhaust pipes of an aphid called the siphuncular, and also there's the cauda, the tail. You'll see an aphid being born here, and above that aphid is a tail, the cauda. Let me just show you in more detail what they look like. The chorda can be various shapes and sizes. So we've got long pointy ones and broad plate-like ones. And that's diagnostic, as well as these exhaust pipes, or cornicles, as we call them in the UK, siphunculæ, as I sometimes call them, which is an American terminology for those which produce some of the waxes, for example, that you can see on these, and other chemicals, by the way, on these aphids. So the challenges are that if you want to do anything small and blobby and just look down at that Petri dish, I mean, computer models will have a nightmare trying to decide what that is. They lack charisma, they lack color. There is lots of phenotypic variation. That is, just like with other insects, they have a sort of Jekyll and Hyde color variation. They can be green, pink, or purple within the same species. They can have wax, not wax within the same species. But also their presentation and orientation in the Petri dish, not necessarily the same. Therefore, models have to learn their new orientation or presentation to get an accurate ID. Importantly, the detritus and the crowding of those aphids in the Petri dish really matters. I'm going to show you something really quite interesting now. So work with University Passo Fundo and Embrapa in Brazil on deep learning models to identify these tiny little aphids is underway. And what we've done is we've tested the models with sample assays from the insect survey. And we've got parasitoids, which look a bit like aphids if you're naive, a computer naive model, but they aren't, of course. And we've also got detritus. And in the top slides, we've kept things simple. We've got either one aphid, one parasitoid, and we've arranged them and confused the model with things getting in the way, if you like. So the first top three, well, they can say fairly accurately, yep, there is at least one aphid there. They're not 100% precise. They do make mistakes. But the mistakes really come when you get a whole mass of aphids. Now, we've been kind to the model in the bottom left-hand slide, because what we've done is actually separate all the parasitoids and aphids into separate components. But as we go along that series, we then confuse the model with detritus, and then we confuse it more by making the aphids overlap. And just look what happens to the deep learning model. So those boxes around are IDs that it's making. And then you can see, actually, it's not bad. It's not amazing, but it's not bad. But at the very end, we've got the same number of aphids, the same number of parasitoids in that bottom row. They're just arranged differently. How the models completely underperform when those insects are overlapping. And that's because they associate that overlapping of the insects with a new species. They say, I can't possibly identify this. I don't recognize this. That's a real problem for small insects and deep learning. We try to tackle another problem is can you hear aphids in flight? You can't, they're silent. But I can challenge you all, and probably I would expect that all of you, even if you had your back faced to a buzzing wasp or a bee or a mosquito, you could identify it without looking at it. And that's because of the harmonic resonance in the wings. Just like a Stradivarius violin that has its own sound due to the wood that it's made of. In the same way, aphids have, and all other insects have their own sound, because these aphids are constructed just slightly differently compared to another species. And so along the bottom, we used light, unbelievably, transformed into a sound wave to then say something about the wavelength or the harmonic frequency of those aphids. And the closely related aphids on sycamore are a little bit different in their signature, but fundamentally different from the beetle on the far right-hand side. And so we're using that information to try and get more accurate IDs with a view of deploying such a sensor in the field. On everybody's lips, is there an insect Armageddon? And you've got opinions left, right, and center. But I want to highlight a really important piece of work by Kelvin Conrad, who sadly passed away. And he published a paper a long time ago. You can see 2009 here, where he actually highlighted the insect biodiversity crisis. Now, you may all know about the Hellman paper that came very much later, but Conrad saw it first. And even before him, there were others who actually were able to describe declines, but not actually quantify them. comrade using our long-term data did exactly that. And then Chris then followed to look at abundance changes in flying insects, particularly flies in our traps, to show how that was changing over time, again, reinforcing possible change. So in collaboration with Dan Bloomgard, who was a PhD student with me in the insect survey, and Rosa Menendez at Lancaster, Dan and Chris Shortall, who I've talked about earlier, I wrote a paper on the standardized data set that we have for the survey for estimating whether or not insects were declining, using the moth data and also the aphid data to say something about insect decline more generally. Now, before I go on to a little interpretation, I just want to highlight the work of Ishbel Hayes, who is handing in her thesis tomorrow on the flight-to-light response to the decline of Britain's moths. She's taken satellite information, created a new driver data set, and also looked at responses between dark and light sites across our network to show that light pollution is really having an effect. Now, remarkably, light pollution is increasing year on year across the globe by around 10%. So it's a significant driver now. And what we were able to show was how the differences were emerging between farmland and woodland, for example. Woodlands were in steep decline and farmland looked like it was varying, but on average across time, it wasn't declining at the rate that woodlands were. So we see those two groups there, and then we see the trends overall. So moths, just like they have in the State of Britain's Larger Moths report, declining just over 30%. And we can see some variation there in the non-linear model, but it's a decline all the same. And we see the aphid trends too. Now, remarkably, think about this. Think about the millions of tons of insecticide we've thrown at these insects, and yet they're not declining. So there's something about the resilience of those, and I kind of respect aphids for that. So we've taken a huge amount of data described above to say something about the aphid trends, but they're not moving, they're not budging. So Dan then picked up the baton from my paper that I wrote as first author to his first author paper, and working with Mark Botham at CH and Rosa Menendez, he concluded that broadleaf woodlands was the only habitat in which we saw all four metrics of decline heading really the wrong way. 51% for abundance, 52% for biomass. Species richness was falling 14%, and species diversity around 15%. And that what was interesting was that these moths were actually stable until the 1980s, but then saw a rapid decline. And Dan took deer data set to understand whether or not the host plants were removing those the deer were removing those host plants, but we couldn't find evidence of that. So as yet, we don't know why that's happening. But there is some good news that actually some moths are actually benefiting from some of the policy changes that we've made as a nation. And I will give you a sort of an upbeat response, work highlighted by Douglas Boyes, who again is sadly with us. What a great scientist he was, but he highlighted that actually things like the footman moths that feed on lichens were increasing more than 1,000% because we had made policy to change the air quality. And that air quality was improving the opportunities for lichens, and as a consequence was improving the opportunities for footman moths. So some great news to be had, obviously some sad news too. Again, just like to say how we use our data. Of course, the state of nature, the atlases that are being produced, but also feeding into the Office of National Statistics about habitat and ecosystem changes, and trying to have a policy response to insect declines through the Parliamentary Office for Science and Technology. And you can read that online if you want to. Also look at evidence in Hansard, where moth decline is discussed. And our work is cited also in the Intergovernmental Panel on Climate Change too. So a lot of work that we do is really important to inform the debate. So I'm going to move on to my last topic here, which is neonicotinoids, which will be no doubt very familiar to you all. And it's a sensitive subject where growers, farmers of sugar beets are contracted to grow sugar beet for us to eat as food. And remember that it's us who are eating the 34 kilograms on average per annum, demanding that crop be grown. And as a consequence, the major vector Mysis persicae, the peach potato aphid, the vectors virus yellow is a complex of 3 viruses that can completely wipe out sugar beets, as it did in 2020. Now, in 2020, we had left the EU. The EU had already mandated in 2018 that neonics could not be used any further. But the damage was so severe that in 2020, some farmers lost 100% of their crop. Now, when I say 100%, so what I mean is that some farmers in Cambridgeshire lost £600,000 just for sugar beet alone because of this one aphid. It's a serious, serious problem. Now, we forecast that virus yellow's incidence will be greater than 80%. On average, across the whole region, it was 38%. But we have that variation. Of course, it's one figure trying to capture that spatial variation. So in 2021 and 2022, using our long-term data, we then forecast virus yellows for the industry and for DEFRA. We didn't make a decision on whether or not neonics should be coat seed rather. We just provided the evidence based on our forecasting models as to what the likely virus yellows incidence would be. Now, you've got to remember, we're doing this in the 1st of March, when no aphid is flown, when no seed has been planted, and when no one knows 6 months ahead what's going to happen in the field. That's probably one of the major forecasting challenges anywhere in the world. And so getting it right is really, really, really very hard. And so in 2021, we forecast 8.3% of the crop would succumb to virus yellows. The trigger that DEFRA issued was 9%. Our value was under that, and therefore Cruiser SB, the neonicotinoid, was not permitted. And on inspection of the crop, at the end of harvest, it was around 2%. That's pretty good modelling. In 2022, we forecast a value of 68%. The trigger was 19, and the neonics were permitted, and some of the growers opted in. But now there's a resurgence of growers who are not necessarily taking up neonicotinoids, but choosing another course, and that's got to be welcomed. We didn't know the national instance of the field because I referred to earlier the beet moth came across the continent and really affected the crop, as well as the drought that then was found. And actually, those cause either loss of leaves to be sampled as assays or cause the crop to go yellow, which looks a bit like virus yellows. So what we're dealing with is a really complicated circumstance of transmission. I'm very briefly describe a sugar beet crop in the middle, and we've got aphid vectors flying around this landscape, some up to 100 kilometers, but the great majority locally, probably between fields, not between landscapes necessarily. We've got some plants that we love in the field margin, such as speedwell and dandelion, being virus reservoirs that the aphids can feed on and collect, if you like, the virus to then transmit it in the field. So we're testing that transmission pathway in a project called Molecules to Land. We're funded by BBSRC. So we then deploy a model to try and predict virus yellows. And you can see the heterogeneity across East Anglia where sugar beet is grown of virus yellows incidence, particularly around the wash, there's very high incidence. But we try and predict what that is using our first flight information. So the first aphid to arrive in the trap, we predict that. We then predict the abundance to mid-part of June, and then use a modelling environment to say something about how the virus will spread and the impact on the crop. And then using that information, DEFRA make a decision. Here's a rather compelling model about the first flight or the first individual recorded at Rothamsted over the last couple of decades, since 1965. And all it says basically is the first flight of the aphid is affected by temperature, the Jan-Feb mean temperature. When it's very, very warm, like between 6 and 7 degrees, aphids will fly very, very early in the season. And that means that your sugar that you eat, the 34 kilograms, actually will be greatly affected. So we may need to actually use some sort of form of mitigation. I want to say about the super El Nino effect coming this winter means that we're probably going to go beyond this. We're going to have record levels potentially of first flight arrival. In fact, the season may never end. We may be going to a sort of subtropical flight season for this aphid, which will be a new kind of agriculture. So watch out for that space. Here's some of the variation that we see every year in the flight response between the 1st, the 5th, the 10th, and the 20th catch of an aphid at a site. You can see that there's no real pattern. So it just justifies our work in doing the forecast every year and assuming nothing about the prevailing season. So we have a time series of virus incidents. And you can see that with the blue dots. Generally speaking, between 1996 and 2018, when everyone used near-NICs, there was no virus around. The orange line says what it would be if we didn't have near-NICs, by the way. And then, so in 2020, we can see the rapid rise up to very high levels of the virus in the crop. So I want to finish with that slide. That's my last topic. I know that I've gone on for quite some time, but I believe it to be interesting to you all. After all, this is the food we eat, and this is the environment in which we live, where biodiversity change are really changing profoundly in our time. But we're going to have our 60th anniversary next year. We had our 50th to mark the significant impact we've had, and we celebrated it with a cake and visitors to Rothamsted Insect Survey. So I just want to finish by saying that I'll be leaving as head of the Insect Survey at the end of the year, and I'm hoping that someone will come along, someone like yourself potentially, to lead the Insect Survey into the new future. And so with that, I'm going to finish. I'm going to pass over to Yoann. So I'm going to stop sharing my screen.
All right, so I will present here an example of what we can do at Rothamsted with the kind of data we collect in the Rothamsted Insect Survey. So in that case, that's training models. Here it's a model that we developed within the DRUID project. That is a project that includes Rothamsted, CEH, Leeds, and Reading universities. And it was published earlier this year in Methods in Ecology and Evolution under the title Explainable Neural Network for Trait-Based Multi-Species Distribution Modeling. And to put it in other words, it's a machine learning approach that will build on trait-environment interactions and is able to derive or explain the environmental filtering and build predictions on that. So we work at the community level and we address community by community as much as many insects as we can across the UK. So far, we've done the butterflies, the moths, Odonata, carabids, Orthoptera, and bees, and more are to come. And so that model is, it's called trait-based because it has two kinds of inputs. One is the environmental drivers. They are, in our case, very numerous. We focus on a very agnostic approach where you deliberately put a lot of drivers inside it. Basically everything you can put your hands on and that you think is remotely relevant to insect presence, you put them blindly in. It's a very different kind of approach than statistics would call for. And in our case, we have drivers that are land cover derived, for example, the proportion of coniferous woodlands, or some weather derived, like maximum temperature reached in the year, insecticides, elevation. We have maybe between 45 and 55 drivers, depending on the community. And the other input we have for this kind of modeling is the species traits. They are key to our approach. Here it's 6 examples of Odonatas, for which we have traits. In this case, the traits are body length, breeding habitat, flight length, number of habitats, volatinism, this kind of stuff. And we have that informed for the whole community. So in that case, for the Odonata, that is, I think, 34 species across the UK, for which we also have, on the other side of the equation, observations. This for the Odonata are presence only, so that comes with difficulties. But for example, they can also be presence and absence, like for the butterflies and the moths from the Rothamsted Insect Survey light trap network. When we have presence absence, it's much easier to train the model and make better predictions, but we can also work with presence only, with the limits I will talk about soon. And the output is the kind of map, species-specific map you see on the right, sorry. Yeah, and we train the model to reproduce the patterns of observations we see, and then, if you want, interpolate across the UK to get probability of presence or of every species.
Johan, just to let you know, there's a weird noise just started coming from your computer. Oh, yeah, it's stopped now.
I have no idea what it is.
Yeah, no, it's stopped now, so that's fine. Carry on.
Okay, maybe it's because I stopped talking. So at the heart of our approach are the species traits. Those are often tricky to get, but especially when you have very big communities like the moth. But for example, for the butterflies here, we have 7 traits that characterize the different species. So every species is located in that 7-dimension space, and it's here projected in the 2D space. So species that are alike are shown as neighbors in that plot, for example. The use of traits is often seen now in species distribution modeling, because in theory you would expect some species that are alike to react similarly, to respond similarly to some drivers. So that comes with exceptions, of course, but that can help the model build decent predictions for species that are very poorly recorded. Those species can usually sort of piggyback on their neighbor species that are often more better recorded. Another use of the traits is for explaining what is happening with our model. Here, for example, you see for the mouse, what we have here is what we call the force corner matrix. That's characterizing interaction, positive or negative, positive in red, between the traits we entered and all the drivers we have. So there's a row here, the row intercept, that is easy to understand. It is a non-trait mediated effect, so an effect that applies broadly across the community, and that is not mediated by traits. And here, for example, we highlight big effect for the moss, a very strong positive effect of the presence of woodlands, especially conifers, for the presence of moss in general, and maybe a negative effect from the presence of wind in general. When we go a bit above, we have the trait environment interaction. Sometimes they're a bit more tricky to wrap your hand around, but here, if we take this one, it's positive. It's between arable and the number of host plants. So it's basically saying that the effect of arable land is more positive to species with greater number of host plants. So that's kind of saying that generalists are a bit more suited to agricultural environment, and something we know about. At the species level, we can then derive niches. For example, that species here is positively affected by what is here temperature diurnal range, but this one is more likely negatively affected. So that can be simpler to understand, and in some case, depending on the species, be useful. For our modeling approach, it can also be useful to drastically reduce the problem by dropping irrelevant drivers once you reach that point. A lot of them are never highlighted as key to the prediction, so you might as well just drop them, make the problem much simpler, maybe reduce to 6 or 7 drivers that are very relevant. And once you reach that point, maybe other statistical methods that are a bit more computationally demanding may become tractable, like, for example, HMSC, that a lot of you might know about. So that's for the explanation. We have a difficulty that we've reached once we passed the butterfly and moth data, which are exceptional data sets, is working with presence only. Presence only data result from usually from opportunistic sampling, where an observer will declare the presence of one species at a location, and that declaration does not mean the absence of all the other species of the community of interest. But it is something that we have to regard as the absences of the other species to input that data in our model. So that brings a tricky, it's quite a tricky problem, because we are able to assess the skill of our model to predict presences correctly, but never its absences. So in some way, the problem can be regarded as trivial, and we can never be sure that our model is predicting absences accurately. So for one way of checking that is what you see here is a simple sense check that you overlay the observations with, so in black points are the observations since 2010, and you overlay it with the prediction map for 2020, and you see if it makes sense. But it's very circular, and it's a very weak way of testing the quality of your model. So to go beyond that, we have built an online tool that some of you might help us with. If you're an expert in identifying some of those insects, so those 3 communities there, and also the bees, and are able to locate maybe some of those, please print screen now and follow that code there. Can help us. It will bring you to a survey. If you click here on Orthoptera, for example, you will get a survey about Orthoptera in which you can select a species that you think you're able to identify. You can do it for only one species if you want. Then you're shown 10 random points across the UK. If you're not happy with those, you can reshuffle those. And then you answer only for the points you can answer for, basically. If you're from the Southwest and can answer for that, you can zoom, obviously, and get details of the location you're asked about in terms of drivers. And there you enter a value between 0, stating that you're certain that that species is absent from that area, and 100, that says you're certain it is present. And everything in between is there to quantify your certainty on that. And you can just leave most of the table empty and just give one answer here and there. And that would anyway be of great help to us. So thank you for that. And thank you for your attention, because I'm finished.